Hello friends, in this video we will discuss the paper of Jamia Millia Islamia of MSc Biotechnology. This is part 2 of the video in which we will discuss the other 50 questions. So let's start. Now the question is, protein synthesis takes place at option A ribosome, option B nucleus, option C nucleolus and option D mitochondria. As we know, nucleus and nucleolus are involved in transformation of genetic material. Then mitochondria is used in ATP synthesis. So option A is the correct one, that is ribosomes that helps in protein synthesis. Option number A. Compounds which is added to soap to import antiseptic property is A. Sodium lauryl sulfate, option B. Sodium dodecyl benzene sulfonate, option C. Rosin and biothianol. The correct option is option number D. Biothianol. So what happen is it is used to make the soap to import antiseptic property to it. All soaps are made by boiling fats or oils with suitable hydroxides. But what happen is the sodium lauryl sulfate, sodium dodecyls are anionic detergents. They are also added, but they do not impart antiseptic property. Similarly, rosin is also added to soap, but to make it leather will. So the correct option is option number D, biothianol. Now, an antibiotic contain nitro group attached to aromatic nucleus is A. Penicillin, B. Streptomycin, C. Tetracycline, and D. Chloramphenicol. So let us look at the structure of chloramphenicol. It contains nitro group which is attached to the aromatic ring. So the correct option is chloramphenicol, option number D. Now, the number of chlorine atoms in biothianol is, so let us see the structure of biothianol. Here we see there are two aromatic rings connected by sulfur, multiple fluorine atoms, that is 1, 2, 3 and 4, and two hydroxyl groups. So the correct option is, if we see only chlorine, it is 4. Correct option is option number B. Which of the following gene is called guardian of genome? A. P53, B. P450, option C. Cytochrome, and option D. Homeotic genes. So the answer is A. P53. How? Because P53 protein encoded by TP53 gene in humans, that is known as guardian of genome. Why? Because of its activities. Because it maintains genomic stability through the repair of damaged DNA. Also in integration of cell birth or death signaling pathway with the DNA damage checkpoints. So option is option number A, P53 proteins. Now, homeotic genes are responsible for. So what happen is, these homeotic genes are the genes which regulate the development of anatomical structures in various organisms such as echinoderms, insects, mammals, and proteins. These genes encode a transcription factor proteins, and these proteins then affect the development of the regulating downstream gene network involving in body patterning and development of anatomical structure. Option number A is correct. It is not related to homeostasis or cell cycle regulation or adaptation to stress. The correct option is option number A, developmental of anatomical structures. Now, primary acceptor of CO2 in photosynthesis is A is ribose, B ribulose 5-phosphate, C ribulose 1,5-bisphosphate, and D 3-phosphoglycerate. So C. This is the C3 cycle, which we can also say Kelvin cycle, dark reaction. In this, what happens is the CO2 formed in light reaction comes and is taken up by the RUBP. So acceptor is RUBP, that is ribulose 1,5-bisphosphate. And then further the steps, that is carbon fixation, reduction of the 3-PGA formed, and the regeneration of RUBP takes place. These are the 3 steps of Calvin cycle. Okay, so they have asked about the primary acceptor of CO2, that is ribulose 1,5-bisphosphate, option number 3. Now the question is, the organelle of C3 plants where glyoxylate is formed in A. Ribosome, B. Peroxisome, C. Mitochondria, and D. Nucleus. So in this picture, we see that there are 3 organelles: chloroplast, peroxisomes, and mitochondria. And the formation of glyoxylate occur in peroxisomes. That is option number B, peroxisomes. Now, cytokine synthesis is maximum in A. Roots, B. Leaves, C. Shoot tips, or D. Fruits. So cytokinin is a plant hormone. It promotes cell division. And what other functions are? It delay the destruction of chlorophyll and protein. It delay the senescence in leaves, and it is mostly produced in root apex. So the correct option is option number A, roots. Now, gibberellin treatment to grape plants leads to. So as we know, gibberellin is your phytohormone. Option A, fruit ripening. Option B, seedless fruit. Option C, cell division. And option D, leaf fall. So what happens is, if gibberellin treatment is given to the grape plants, it leads to the formation of seedless fruit. Seedless fruit, that is, we also known as parthenocarpy. However, fruit ripening, cell division, or leaf fall is not initiated by gibberellin. So option number B is the correct one. Now, the movement of chrysanthemums is due to A. Phototropism, B. Humidity, C. Oxygen depletion, and D. Heat sensitivity. So the movement of chrysanthemum is not related to humidity, oxygen depletion, or heat sensitivity. Our correct option is option A. Phototropism. And due to this, they thrive in intense sunlight of summer. So our correct option is option number A. Phototropism. Now, solutions of quadratic equation x square plus 5x minus 6 is. 4 options are given to you. So for this, we know the technique of middle term splitting. We will put this equation equal to 0. Now split the middle term. We get 6x minus x. Taking common from x square plus 6x and minus x minus 6 से. So यहाँ से common x and अंदर बच गया आपके पास x plus 6. Similarly यहाँ से आप common लोगे तो minus 1 will be common and x plus 6 will be in the brackets is equal to 0. So our one pair is x minus 1 and one pair is x plus 6. x minus 1 को 0 के इक्वल रखेंगे तो 1 will be x is equal to 0 1 and another x plus 6 को 0 के इक्वल रखोगे तो x कितना आ जाएगा minus 6. तो आपकी एक वैल्यू 1 आएगी और एक वैल्यू minus 6 आएगी. तो वो आपकी कौन से इक्वेशन में है, क्वेश्चन में है, ऑप्शन में है, ऑप्शन नंबर बी, x is equal to 1 and x is equal to minus 6 is the correct one. Now, pollen tubes lack A. Mitochondria, B. Nucleus, C. Plastid and D. Cell wall. So pollen tubes हमें पता है यूनिक स्ट्रक्चर होते हैं, हमारे फ्लावरिंग प्लांट्स में प्रेजेंट होते हैं एंड दे हैव सर्टेन फीचर्स जो उनको डिस्टिंग्विश करता है टिपिकल प्लांट सेल से. So they contain mitochondria for their cellular respiration and nucleus for genetic material and regulating cell functions. But they do not have plastid like chloroplast. So option number C is the correct one. Now, nucleotide is made up of option A, nucleus plus DNA, option B, sugar plus phosphate, option C, base plus phosphate, and option D, sugar plus phosphate plus base. So let us see the structure of two molecules which can be asked other than nucleotide is nucleosides. So we should know about both. In case of nucleotide, we have a sugar. This is our sugar. The base can be adenine, guanine, thymine and cytosine in case of DNA, and adenine, guanine, uracil and cytosine in case of RNA. Similarly, if sugar they are talking about, so in DNA we have deoxy sugar and in RNA we have ribose sugar. Other than this base and the sugar, a phosphate moiety is also attached in case of nucleotide, whereas in case of nucleosides there is no phosphate attached, only the sugar and base is present. So If they are asking about nucleotide in our question, so our correct option will be option number D, sugar, phosphate plus base. Now, our question is, crossing over takes place या occur during A. Mitotic metaphase, B. Mitotic anaphase, C. Mitotic prophase, D. Meiotic prophase. So what is crossing over? Crossing over is exchange of genetic material between two homologous chromosomes. What happens? These homologous chromosomes firstly get aligned. Okay, the blue one and red one, these are the homologous chromosomes which firstly align. After that, crossing over takes place and chromosome crossover are formed. This X-shaped structure which is formed here is known as chiasmata. Chiasmata formation takes place, and after that the recombinant and non-recombinant chromosomes get formed. And this event occurred during meiotic prophase 1. That is option number D is the correct one. Excess of nitrogen leads to A. Elongation of roots, B. Reduced stem strength, result in lodging, C. Yellowing of roots, and D. Widening of leaves. So what happens when there is excess of nitrogen in the level and the basal nitrogen ratio? The basal internodes become slender and fragile with a thick stem wall. While filling degree chemical components and the strength of the stem decreases gradually, which significantly increases the lodging risk. And if we talk about what is lodging, so it is the displacement of stem and roots of plants from their proper and vertical placement, either due to high ion applications, that is nitrogen application, or high wind speed, excessive soil moisture, or soil density. So our correct option is option number B, reduced stem strength result in lodging due to excess nitrogen. Now, chlorosis of young leaves is generally due to deficiency of A. Magnesium, B. Calcium, C. Phosphorus, and D. Potassium. So what happened in case of chlorosis? Yellowing of leaves occur due to lack of chlorophyll, and the deficiency of nutrients, that is main if we talk about it, is iron and manganese. As iron is not given in our option, so we will go with the option number A, magnesium. However, it is also caused by deficiency of potassium in some small amount, but magnesium is the major cause. So option number A will be the correct one. Other than that, chlorosis is also caused by soil pH level, ozone injury to the sensitive plants, and many more reasons. So our correct option is option number A, magnesium. Now, dehydration of alcohol is an example of A. Redox reaction, B. Elimination reaction, C. Substitution reaction, and D. Addition reaction. So see here, in case of dehydration of alcohol, dehydration means removal of this water. So when we take this dehydration of alcohol, then this acid is used where the formation of alkene takes place and the water is formed. So the correct option is elimination reaction, as there is elimination of the water takes place. Now, melamine plastic crockery is a copolymer of A. HCHO and melamine, that is HCHO is your formaldehyde, B. Formaldehyde and ethylene, C. Melamine and ethylene, and D. Melamine and CH2OH. So this melamine plastic crockery is made up of the monomers of formaldehyde and melamine, that is option number A. And this plastic used for making crockery is melamine. That is because melamine resists fire and it can tolerate heat better than other plastic. So our correct option is option number A, formaldehyde and melamine. So A is the correct one. Now in bacteria, sexual reproduction takes place by A. Budding, B. Binary fission, C. Transduction, and D. Transformation. What happens is in bacteria, however, sexual reproduction does not take place, but the genetic exchange occur by the method transformation and transduction. So C and D can be correct options. Which of the following ecosystem is unique to India? A. Mangroves,
B. Cold desert, C. Meristic swamps, and D. riparian forest. So, mangroves are the unique type of coastal ecosystem found in tropical and subtropical regions. Also, they are dense forests of salt-tolerant trees, shrubs that thrive in intertidal zone where land meets the sea. So, option number A is the correct one, mangroves.
Now, which is the latest influenza outbreak in India prevailing in 2023? Option A H3N2, option B Omicron, option C Delta, and option D H7N7. So the latest was H3N2. It is a subtype of influenza, a virus that has been associated with more severe illness and high mortality rate among children, older adults, and those with underlying health conditions. And this H3N2 is responsible for most influenza-related hospitalization and deaths globally according to World Health Organization. So our correct option is option number A, H3N2. Now, our question is benthic organisms of lake or sea are usually A. Producer, B. Herbivore, C. Carnivore, and D. Decomposers. So, what are benthos? The community of organisms जो कि benthic zone में present होती है उनको हम benthos बोलते हैं and इसमें हमारे invertebrates आते हैं that is crustaceans, amphipods, polychaete worms, snail etc. and these all are decomposers. So our correct option is option number D. Now, maximum diversity occur in A. Tropics, B. Equator, C. Temperate, and D. Poles. So, as we know, species diversity decreases as we move from equator to poles. So, according to this, tropics have more diverse species compared to temperate, polar regions or equator regions. So, our correct option is option number A, tropics. Now, which of the following technique is most efficient to investigate RNA genome viral infection? A. Southern blotting, B. Northern blotting, C. Microarray, and D. RT-PCR. The correct one is D. RT-PCR. It is reverse transcription polymerase chain reaction, which is a laboratory technique that combines reverse transcription of RNA into DNA and the amplification of specific DNA targets using PCR, that is polymerase chain reaction. And this is used to investigate RNA genome viral infection. So correct option is option number D.
Our question is, in sandwich ELISA, the molecule captured is option A antibody, option B antigen, option C enzyme, option D nitrocellulose membrane. So what happens in sandwich ELISA is, first of all, the antibody is captured. Second step, antigen is joined to the antibody by some interactions. Then we add labeled antibody that can be Written labeled or labeled by any probe or something else. Then we add our enzyme linked to the substrate and a colored product is formed. So they have asked the molecule captured is. तो capture हमने किसको किया है? Antibody को capture किया है sandwich ELISA में. So your correct option will be option number A, antibody. Our question is the most likely method used to determine the structural details of cell organelle is A. Autoradiography, B. Microdissection, option C. Electron microscopy, and option D. Phase microscopy. So the correct one is electron microscopy. How? Because electron microscopy एक ऐसी टेक्निक है जिससे आप हाई रेजोल्यूशन इमेजेस बायोलॉजिकल और नॉन बायोलॉजिकल दोनों स्पेसिमेंस को स्टडी कर सकते हो. ठीक है. It is used to investigate detailed structures of tissues, cell as well as organelles and macromolecular complexes. Other than this, it is also used in study of microorganisms like bacteria, virus, and other pathogens that have made the treatment of diseases very effective. So the correct option is option number C, electron microscopy. Which of the following helps in osmoprotection in plants? A. Proline, B. Tryptophan, C. Glycine, and D. Histamine. So proline is used by the plants in osmoprotection. How? Because it serves as a compatible solute and as it is a molecular chaperone, so proline act as an antioxidant also and serve as an ROS scavenger. ROS means reactive oxygen species scavenger. So the correct is option number A, proline.
Which of the following is likely to be absent in normal frog ECG? A, P, B, Q, C, T and D, R. So for normal frog ECG, Q wave हमारी absent होती है. That is normally responsible for depolarization of interventricular septum. So correct option is option number B. बाकी आपके पास P, T and R waves present होती है.
The main function of hypothalamus is A. Thermoregulation of body, B. Thyroid secretion, C. Bile secretion and D. Control urea concentration of body. So the main function of hypothalamus is thermoregulation of body. It means it maintains homeostasis of all the things in our body. So option number A is the correct one. 2-aminopurine induces mutation by A. Base pair change, B. Frame shift, C. Duplication and D. Deletion. So what happened is 2-aminopurine is a base analog which cause transition and frame shift mutations in E. coli or other organism. And how it does so? It mispair with cytosine and various other mechanism and cause the frame shift and transition mutation. As transition is not given in our options, so frame shift is the correct one. That is option number B. The compound used in preservative for food products such as tomato ketchup and fruit juice is A. Sodium salicylate, B. Sodium benzoate, C. Sodium acetate and D. Formic acid. So sodium benzoate under certain condition is converted into benzoic acid, which is bacteriostatic, and this benzoic acid is then absorbed into the cell and the intracellular pH falls to 5 or below. So option number B is the correct one. Now dry ice is A. Solid NH3, B. Solid SO2, C. Solid CO2 and D. Solid N2. So what happens is dry ice is solid carbon dioxide. Okay, it is known as dry ice because it goes from solid to gaseous state without going to liquid phase by the process we call as sublimation. So the correct option is option number C, solid CO2.
Triticum aestivum is an example of allopolyploid, autopolyploid, chromosome doubling, autoallopolyploid. So, Triticum aestivum is wheat. That is an example of allopolyploid. Now, what is allopolyploid? It is basically an individual that is a hybrid of two different species and possesses more than two set of chromosomes. So, option number A is the correct one, allopolyploid. Holiday junction is observed during A. Mitosis, B. Interphase, C. Recombination and D. DNA damage. So holiday junction एक structure बनता है that is during recombination जब हमारी crossing over होती है. That is the feature of our meiosis. Okay, so basically it is formed during or observed during recombination. So our correct option is option number C, recombination.
Now see, pigments used in classification of A. Algae, B. Lichens, C. Bryophytes and D. Pteridophytes. So algae are classified into following groups based on their photosynthetic pigments. For example, chlorophyceae are the green algae which contains the pigments like chlorophyll A, chlorophyll B and zeaxanthin. Phaeophyceae, brown algae that contain chlorophyll A, C, beta carotene and xanthophyll. So the correct option is option number A, algae. These are classified on the basis of pigments. Now, various plant diseases are caused by organisms of size lesser than 1 mm and they lack cell wall. The organism is A. Virus, B. Bacteria, C. Mycoplasma and D. Fungi. So the correct one is Mycoplasma. They are less than 1 mm and they lack cell wall. The various disease which is caused by Mycoplasma in plants are, which is broom of woody plants, coconut lethal decline, grapevine yellows, apple proliferations. These all are caused by mycoplasma. So the correct option is option number D. Question is, which of the following enzyme is responsible for making a DNA copy from RNA? RNA से DNA copy कौन सा enzyme बनाता है? A. Reverse transcriptase, B. DNA polymerase, C. RNA pol 1 and D. RNA polymerase 2. So here, see in this picture, a replication occurs, so DNA is formed. DNA to RNA is formed by transcription in which RNA pole 1 and 2 is involved. So C और D को हम neglect कर सकते हैं. These are wrong. DNA polymerase जो है आपका replication में use होता है. तो आपका बचा कौन सा? Reverse transcriptase. That is used for making a DNA copy from RNA. Okay. See here, the reverse is reverse transcriptase. RNA से DNA को बनाना is by the help of reverse transcriptase. Option number A is the correct one. Now, a pentamer immunoglobulin is option A IgG, option B IgA, option C IgM and option D IgE. So, if we see the structure, it is a structure of IgM, which is a pentamer. 1, 2, 3, 4 and 5 forms the pentamer that are connected by the joining chain, which we also known as J chain. Disulfide bonds are also present and heavy and light chains present. So if we talk about pentamer, so it is IgM. Other than a pentamer, it also acts as the first line of host defense against the infections and also provide the activation of complement system on binding to the antigen. So our correct option is option number C, IgM. Now, which one of the following viral vector has the large capacity for foreign DNA with respect to gene transfer to animal cells? Option number A, adenovirus, B, retrovirus, C, baculovirus and D, coronavirus. So, the correct one is adenoviruses. They are non-enveloped, 80 to 120 nanometer in diameter होते हैं, have a linear double-stranded DNA genome जिसमें 30 to 40 kb का insert हम transfer कर सकते हैं. So, for gene transfer vectors, they can carry a fragment which is very large of the foreign DNA. So, adenovirus is used. The correct one is option number A, adenovirus. The term single cell protein SCP was coined by A. Louis Pasteur, B. Roger Mendel, C. Carroll L. Wilson and D. Ian Wilmot. So Professor Carroll Wilson created the term single cell protein in 1966. What is single cell protein? It represents microorganism biomass or protein extract that can be used in animal and human nutrition. So the correct option is option number C, Carroll L. Wilson. The coefficient of variance can be calculated by using following formula. 4 options are given to you. The coefficient of variance is calculated by dividing standard deviation by either arithmetic mean or geometric mean depending upon the context or nature of data given to you. Okay. So in our options, 2 options can be correct, that is option number A and option number D, because they have not mentioned the data in which context we are using. So A and D can both be the correct one. However, if we see, then if you are working with data that represent measurement on ratio scale, then we use arithmetic mean, and if your data is logarithmic scale or the exponential growth, then we use the geometric mean. So A or D can be correct based upon the data which we use. Now this question is related to combination. So if this value is given to you, you have to calculate the value of n. Option A 12, option B 14, option C 16 and option D 18. So by using the formula, we calculate n minus 6 is equal to 12, where when we solve this, we get n is equal to 18. Correct option is option number D. Thank you friends for watching our video. Please like our video, share our channel and also subscribe our channel for more updates.